Hi, I'm Professor Jean-Paul Desaulniers. I'm a chemistry faculty member here at Ontario Tech. I do research in RNA, looking at RNA chemistry and RNA biology, and I also teach courses in chemical biology and organic chemistry. So, as a young child, I was always fascinated by biology and biochemistry. I was really interested in how molecules interact in the body. It wasn't until university that I realized that I actually really enjoyed chemistry, and I was really interested in how chemistry intersects biology. Now, what is chemical biology? So chemical biology describes chemical processes and molecules and how they can interact and how they can manipulate biological systems. So what have I learned with chemical biology? Well, I've learned that you can take biological macromolecules like RNA, for example, and you can use chemistry to modify it in order to improve its properties and its biological function. So at Ontario Tech, we have a lot of great energy. The students are excited about their projects. And we have a very, very nice collegial feeling where students can reach out and work with each other, faculty work well with each other, and it really creates a nice environment where we're not competing with each other, but we're working together to move science forward. Enjoy the ride. I know some days might feel long, but it is a relatively short time you'll be here at Ontario Tech. Have fun, learn as much as you can, learn to work with others in your field, but also learn to work with others who might be outside your discipline, and being able to communicate with experts and non-experts is a very, very valuable skill. And this will help build your network and will help you in the long run to help achieve your goals. But my favorite topics to teach are chemical biology and organic chemistry. At Ontario Tech, I teach chemical biology, and we look at the synthesis of how we can make peptides and oligonucleotides chemically in the lab. And this is very eye-opening for students because for the first time they can see how we can make these molecules in-house and for various biological applications. Furthermore, by using chemistry, we can learn to modify these biological macromolecules like peptides and oligonucleotides to further improve their properties for downstream applications. I also love organic chemistry, and I love learning about reactions and the fundamentals of organic and teaching those fundamental rules for things like various downstream reactions. So as a researcher now, I've learned to realize that it's very normal for experiments to fail, actually quite often. And it's something I think maybe a lot of students don't realize. They think that because an experiment didn't work one afternoon in the lab, that the onus is on them. In fact, this is quite normal. And the other thing I would recommend for students is to get to know as many people as you can. Collaborate with your classmates, introduce yourself to professors, and start your network early. And moreover, have fun and reach out when you need help. So the heart of my research program is centered about understanding gene expression. And what we know is that aberrant levels of gene expression is a hallmark of many diseases such as cancer. So I work with a class of molecules called RNA, and specifically we're working with gene silencing RNAs that can turn off the expression of these particular genes. In our group, we use chemistry to modify these RNAs, which we call short interfering RNAs, or siRNAs for short, in order to improve their properties. We want to improve their stability so they can last longer in the cells. We want to modify them with special groups so that they can get across the cell membrane better. And we're trying to control their potency. One way that we control their potency is by employing photoactive groups within the RNA to turn the RNA on or off. So by using different colored lights, we're able to turn the RNA on and make a gene silence, but then we can also turn it off when we don't want that particular activity to happen. Yes, there definitely are other areas of research, but they're very focused on RNA. So one thing that our lab has been working on a little bit recently are a class of molecules called short activating RNAs. And what these RNAs do is that they enter the nucleus and they turn on gene transcription. So they activate, which is in contrast to gene silencing RNAs. It's, however, quite interesting because the double-stranded RNA that can activate is actually compositionally identical to the same RNA that can gene silence. So we're trying to understand the mechanism by which an RNA that is identical in gene silencing can actually get inside the nucleus and activate transcription. The other class of molecules I'm also very much interested in are the mRNA vaccines that I currently don't do research with, but I'd like to someday. But I think mRNA vaccines hold very enormous promise for next generation cures for prevention of diseases such as cancer. Thanks for joining us today. If you'd like to learn more, please check out our website.